I'm going to present you the Gao team work on RGD-dependent response to shear stress in the article published in Biotechnology Progress in 2007. RGD stands for arginine, glycine, and aspartic acid. Proteins with RGD domain binds to specific ligands, as illustrated here with human integrin. RGD domains are known for inducing stress response, but also for their various biomedical applications, such as tumor imaging or drug delivery tools. The work carried out by the Gao team is key in understanding shear stress in plants. They uncover how the mechanisms involving the RGD peptides we are familiar with in animals can also be found in plants. In this presentation, I will first give you the context. We will see that shear stress is a key problem for the production of therapeutic agents with bioreactors. Then, the analysis of the Gao team work will show that RGD peptides inhibit all of the key elements in shear response pathway by interacting with up to 4 families of proteins. And finally, we will analyze the results published since the Gao team work and see a protein called AT14A could possibly be of great importance. Numerous therapeutic agents like anti-cancer drugs such as Taxol have been extracted from plants like Taxus cuspidata, and individual differentiated cells of this taxus can be grown in bioreactors. Bioreactors have been developed to increase the yield of cultured cells and the production of drug. Indeed, they are a high-scale cell culture tool, as you can see here with an industrial bioreactor. Bioreactors use a continuous turnover of the medium, mediated by an agitation system which aims to maintain the culture medium homogeneous. The major issue regarding this technique is the shear stress. Basically, we have contacts between cells with different speed and trajectories. These differences can be noticed also on cellular scale. Both shear stress and mechanical constraints apply on cells. It is worth to notice that an excessive deformation will lead to reduced viability, or even the cell apoptosis. Therefore, it will decrease the production need of the therapeutic agent, explaining our focus on shear stress in plants. Here is the knowledge about shear stress at article time, meaning this scheme is a simplified view of the current knowledge on this field. Lectins and associated proteins will act as the first elements in mechanoperception. Here, the most important point is the role of a putative integrin-like protein. This putative integrin-like protein will activate MAP kinase cascade, which in turn will modulate reactive oxygen species, phenolic concentrations, and alkylation. There is another pathway on which I will come back later, which will associate with wall-associated kinase and EGF. We will now focus on the putative integrin-mediated response. Integrin, MAP kinase, ROS, phenols, and alkylation. Let's sum up. We have seen that what are RGD, what is shear stress, and why is it important. Now we want to know how the Gao team article has characterized RGD-dependent response to stress. The following experiments will use the competitive inhibition. We have 2 actors, natural proteins with RGD domains inducing stress response, and synthetic RGD peptides. Peptides and proteins linked to the same receptors, but peptides doesn't activate the response. So please keep in mind we will observe RGD peptide effects, meaning the opposite of RGD dependent response. RGD peptide will be compared to RGE peptide because of the structural similarity. For all the following experiments, we look at 4 different setups we will represent with the following color code. First one in blue, absence of stress will be the control situation. Second, shear stress only in red, and third, shear stress plus RGE in orange will give similar results. Last situation, shear stress plus RGD in green give an intermediate response. We will first study alkylation, since it is a classical response to stress. Here we have the evolution of pH with time. Without stress, which I repeat is the control test, we observe a constant level of pH around 5.7. With continuous stress only, we have a quick and significant increase of pH, which stabilizes around the near neutral pH. With shear stress and RGD peptides, we have a moderate increase of pH, stabilizing around 5.9. We had stress plus RGE, give an important increase of pH, similar to stress-only situation. Ionization in presence of RGD is significantly less than with the two situations with shear stress, and also significantly more than the situation without stress. These results show that RGD peptide is responsible for pair-short inhibition of the ionization. ROS are a normal byproduct of oxygen metabolism. They play an important role in apoptosis or host defense activation in response to cellular stress. Therefore, the authors focus on ROS production beginning with hydrogen peroxide. We have pictures of representative cells in each condition. On the left, we have contrast photos of Taxus cuspidata cells, and on the right, the corresponding images obtained by fluorescence. Rhodamine was used as a probe to highlight hydrogen peroxide concentration. Rhodamine fluorescent signal stands for hydrogen peroxide production. Without stress, we have a low level of hydrogen peroxide. With shear stress, we have an important concentration of hydrogen peroxide, and with shear stress plus RGD, we have an intermediate response. These results show that addition of RGD inhibits the production of hydrogen peroxide. This is convergent with the previous results of inhibition of extracellular ionization. Altogether, we can conclude that RGD peptides are involved in the ionization response. After that, the authors experimented on the effects of RGD on other reactive oxygen species. They measured absorbance to obtain ROS concentration after the end of the stress. Without stress, we observe a better level of reactive oxygen species. With stress, with or without RGE, we have an important increase in ROS production, and with addition of RGD, we observe an intermediate response. Therefore, the inhibition observed of hydrogen peroxide can be generalized to other ROS. Another classical response to stress is phenol production used by plants to struggle against aggressors. Phenol production appears to be unaffected by the addition of RGE, as all four experiments display similar levels of phenol production. Finally, to understand the link between mechanoperception of stress and all the previous responses, MAP kinase activity is tested. Therefore, the Gao team used a Western blot against phosphorylated MAP kinases. This Western blot detects the 44 and 46 caldidyl MAP kinase, which mediate response to various types of stress. In this experiment, in the first time, cells are submitted to a moderate level of stress, and in the second time, they are allowed to rest. The stress is induced by the rotation rate. Three conditions are tested with this protocol. The first one tests the effects of the sole shear stress. We observe a quick and significant increase in MAP kinase activity for both the 44 and the 46 caldidyl MAP kinase. Second condition tests stress plus RGD peptides. We observe almost no phosphorylation, meaning no activity of the two MAP kinase. Third response tests stress plus RGE. We observe a quick and significant increase of activity comparable to stress-only test. Though, we can conclude that first, RGD inhibits MAP kinase activity, and second, this inhibition is highly RGD specific. So RGD inhibits all of the key elements proposed in the putative integrin response pathway: MAP kinase, ROS, phenols, and ionization. The next step is to identify the partners interacting with RGD. Therefore, the authors used trypsin to fragment these partners when binding to RGD. Then, they analyze and sequence the fragments by mass spectroscopy. The results lead to four families of proteins. First one, von Willebrand factor A dominant proteins are implied in cell adhesion and formation of multi-proteins complex. Second one, collagen, which play an important role to maintain the structural cell integrity. Third one is cell wall protein-like family. And last one, integrin alpha chain, which was already supposed to play a role in stress response. Now, let's sum up the results of the article. First, RGD peptides inhibit stress response key elements. It means that proteins with RGD domains will activate this response. Remember the beginning of my speech. So proteins with RGD domains activate MAP kinases, alkylation, ROS and phenols production. Secondly, RGD partners have been identified. There are von Willebrand proteins, collagens, cell wall proteins, and integrins. And thirdly, we have noticed that inhibition were always partial, which led to the hypothesis of another shear response pathway. Now, let's have a look to the results published for the last 6 years. The overall shear stress pathway is now better characterized. RGD would directly link to lectin receptors and pectin with wall-associated kinase. The putative alternative pathway would have been discovered and used calcium for signal transduction and interfere with the supposed integrin cascade response. Another important point is that the integrin-like protein has probably been discovered and would be AT14A protein. AT14A was first characterized in this Nakpal-Quattrano article 8 years before the Gautim work, but in 2007 it was the only article fully referenced and quoted about AT14A. Unfortunately, still today there is very little literature about it. Nakpal-Quattrano showed that AT14A was located in cell membrane, has transmembrane domain, and has 2 domains with similarities with animal integrins. Therefore, AT14A seems to be a good candidate. Since additional evidences have become available, a BLAST algorithm request on the Unicode database returns at least 2 Arabidopsis thaliana membrane proteins with transmembrane domain and a putative Ricinus communis AT14A protein. Secondly, this study from 2011 shows AT14A as part of the genes activated in response to parasitic stress. And last element, AT14A seems to play a role in cell adhesion and cytoskeleton dynamic, both of them being implied in stress response. But still today, there is a lack of experiments proving the integrin mediating stress response in Taxus cuspidata is an AT14A-like protein. Therefore, I suggest the following experiments. First, make a degenerated PCR. AT14A gene is sequenced, so we can use fragments of AT14A as primers for a PCR. Then, by hybridization, search for an equivalent in Taxus. If found, make both a knockout and an overexpression and see the effects on ROS, phenols, alkylation, and MAP kinase. We could expect knockout to give phenotype comparable to absence of stress, and overexpression to be comparable to high level of stress. Such results would lead us to conclude that AT14A is the integrin-like protein mediating stress response in plants. The validation of AT14A rule could lead to direct and important applications. By silencing AT14A gene, we could obtain plant cells with an improved ability to produce therapeutic agents when subject to shear stress. Therefore, it would be a piece of solution for bioreactor limitations, as evoked at the beginning of my speech, allowing increased production of important drugs. Now, I would like to sum up this presentation. We focused on shear stress and its importance on bioreactor cell culture. Therefore, we have characterized the RGD-dependent stress pathway in plants by putting forward RGD effects and partners. Then, we have summed up the knowledge about plants' stress response. I have suggested a candidate as the integrin-like protein mediating the stress pathway, and finally, proposed an experiment to test it. Thank you for your attention. Do you have any questions?